Hi everyone, my name is Ziv Bar-Joseph and I'm a professor in the Computational Biology Department and also in the Machine Learning Department here at Carnegie Mellon. And I am going to talk today about our efforts and participation in a project that is called HuBMAP. HuBMAP is a nationwide effort led by the National Institute of Health to create a map, a three-dimensional map of the cells in the human body. The NIH has established 13 centers that collectively will work on this project over the next several years. And at Carnegie Mellon, we are leading one of these centers, which is focused on the development of computational tools, or computational methods for analysis of this data, for accessing this data, for visualizing it, and for interpreting some of these results. The overall structure is that some of the centers are going to be focused primarily on collection of information, of experimental work. Some centers are going to work on technology development, and the other centers are the computational ones, which we are a part of, which will focus on making the data interpretable and accessible to the community. As part of this effort, we are going to develop a lot of different computational methods, starting at analysis at very low levels of just normalizing and processing the data that was generated, establishing methods for constructing bigger or larger scale models from this data, and finally making the data and the results accessible to the research community, and also at a later stage to the clinical and community that might be able to use it to make important decisions about specific interventions to treat individuals. The overall goal of the project is to be able to say not just what is the sequence. So the human genome sequencing project was a very important and very successful project, which actually gave us a one-dimensional map of the human genome, which is the set of instructions according to which our bodies operate. But this one-dimensional map is fairly static. It doesn't tell us what happens at different parts of our body. And of course, all cells in our body, there are trillions of cells, are using this sequence, but different cells use it in a different way. And the goal of HuBMAP is to go from this one-dimensional sequence to a three-dimensional map, basically to tell us in which location in the body, and within that location, even sublocations, what are the sets of genes that are used. For example, the brain has hundreds of different types of cells. Some of them are neuron cells, the ones that transmit information and process it. Some are immune cells. A lot of cells are doing other things related to metabolism and so forth. And we would like to know what each of these cell types is doing, where are the different cells located, and how can we determine the set of genes or proteins that are activated in each of these cells. At the end of this project, the hope is to have a three-dimensional map where you can actually zoom in and out like you do in regular maps and determine the things that are activated. And this will open up a lot of possible applications. For example, you can determine what goes wrong in certain tissues or organs by looking at the set of genes that are expressed in those. You can compare healthy individuals to disease individuals to determine what are the differences and what should be treated. We can also look at changes in the genome itself, this one-dimensional map, which we can actually look at the prenatal stage and see if there are any changes, where do we expect to see problems in the adult or in the child that is born, and maybe come up with ways to treat those or develop methods at least to monitor them over time. So this is an exciting project. Again, we are in the early stages. We just got the award, but we are going to work on it. It's a pretty big group at Carnegie Mellon, 4 of the faculty, and our team also includes individuals from Santa Cruz, the University of California in Santa Cruz, from the UK, and from Google. So all of this is going to be part of this NIH project, and we are extremely hopeful that this will lead to a useful map that will be accessible to the research and clinical